So welcome to this presentation of AGBT and Mendel's Pod Live, the future of genome analysis. Welcome to the AGBT audience and to our Mendel's Pod audience, who will be watching this later in the week. I'm Farrell Timpson, which makes me very pleased to be interviewing two leaders of Bionano Genomics, the genome mapping company. Erik Holmlin, CEO, and Alka Chaubey, CMO. Alka, the last time I talked to you, you were doing testing in the lab. Isn't that what you're supposed to be doing?
Absolutely. This is what I'm supposed to be doing, and this is what I will be doing.
Okay. So tell us about why you came to work at the instrument company and what you are doing.
So when we spoke last time, Farrell, I was the director of the cytogenetics laboratory at Greenwood Genetics Center. Mm-hmm. And so everything that Erik has just spoken about, one thing is that structural variations are not new, because structural variations have been analyzed or have been interrogated as a part of the genome since the 1970s. However, the issue was that there is a limitation with every technology that was adopted in the clinic for diagnosis and detection. So structural variations have been around for a very long time. So let me just, add that, in clinical diagnostics. But from there, between then and now, I also, was at another company as the head of cytogenomics, where the focus was to try to use sequencing technologies to interrogate all classes of variations that happen in the genome. So whether it is structural variations or whether it is sequencing variations. So we realized pretty quickly that there are a number of disorders and a huge part of the genome that cannot be accurately interrogated by short-read sequencing. So as part of that, people still need to diagnose and to find the genetic causes of a number of genetic disorders, whether it is cancer, whether it is children with disabilities. And this is where optical genome mapping comes in. So if you remember last time, as from the other side, as a laboratory director, I had been waiting, like my colleagues and peers, for a technology that can be the next generation cytogenomic tool that has higher resolution, that can provide more answers, and can be adopted in the clinic by laboratories that usually adopt newer technologies. So this was the impetus for me joining Bionano to drive this in the form that it can become a valid clinical tool just because of the power and potential that optical genome mapping has.
Okay. So Erik, and by the way, please either of you jump in here.
Yeah, well, I was gonna say, Farrell, that there's another answer to what Alka just said, which is that she grew frustrated by our attempts to do what she described. So she decided to come and push us and really make it happen. And there's no question that she's doing that.
So talk about that frustration and what she wanted to push for.
You're putting me in this situation, okay? So,
well, I'm curious to hear what you want to say, and I'm curious to hear what Erik wants to say about that.
So again, coming from the laboratory side, if you look at majority of the tests that are used in clinical diagnostics, right, by certified directors, by laboratories to offer to the geneticist or physicians or pathologists for, no matter which genetic disorder you choose, there are so many things that we need to put into place, regarding the specimen type, what kind of assay structure should it be, what should be the throughput that a laboratory would want, the ease of adoption, the reproducibility, the fidelity of the assay, and the ability to interpret those variations coming out of any technology appropriately. So Bionano, as you just, started this conversation with the funding and everything, had been moving, obviously, but now we have the leverage to really add a lot of fuel to these efforts to make progress in so many different domains that we can cater to what the customers would want in terms of adoption. So I think this is what Erik was referring to.
Okay, so Alka, have there been LDTs already created?
Absolutely. And prior to my joining Bionano, I actually did validate optical genome mapping for a very unique muscular dystrophy disorder that is very difficult to diagnose. And, a number of very cumbersome technologies are needed just because that part of the genome is refractory to the conventional methods and even to sequencing methods. So this is where, as Erik mentioned, that these long linear molecules come into play, and they have such a huge utility to pick up these large structural variations. So I'll tell you like a little bit of a personal thing that almost 5-6 years ago, when I first saw optical genome mapping and these DNA molecules shooting through the nanochannels, it was one of the most beautiful technologies that I had seen. And this was, at the time when I was still at Greenwood Genetics Center. But this is something that we needed to develop and bring it to this point that these clinical applications can be adopted. Now, keep in mind that if you look at the whole continuum of care, if you start with prenatal diagnosis, right, to try to pick up aneuploidies or other microdeletions in fetuses, right now for prenatal testing, if we move on from there to postnatal, now babies are born, they may have a congenital anomaly, they may have a birth defect, they may have delays and autism, as they're growing. Then we come to leukemias and lymphomas, mostly, that happen in adult onset situation. The whole continuum of care, structural variation detection is the standard of care. And now we have this high resolution technology that can pick up so many things that fall in the limitations of these conventional methods. So there are a number of labs that have adopted and brought on for even a whole genome application. So we have a laboratory that has adopted optical genome mapping in conjunction with whole genome sequencing to have a complete genome test. And then there are other laboratories that have adopted for unique clinical applications. So you can just see that slowly we have made that headway, and now we need to make sure that we can develop more applications.
Alka, do you want to pick up on that and talk to
Sure.
the research discoverers in the audience? Talk about it as a discovery tool, the limitations of sequencing, and what this can open up.
So I think when you mentioned Catherine Brownstein, so I'll just, take it on from where Erik ended. One of the things that she's also part of is the COVID-19 Host Genome Structural Variant Consortium. And that consortium is led by Dr. Ravi Kolhe from Augusta University, and it is an international consortium. We have 70, 80 collaborators there. And using the Saphyr technology, what the main initiative is, because we are in the COVID-19 pandemic, so this definitely requires a little bit of a mention, is to look at the human genome and see what these structural variants are, because we know that the diversity in humans is also largely due to the structural variations in the human genome. So what could be at play, and what could be the predisposing factors, genetic factors, that allow people to have different effects when exposed to the SARS-CoV-2 virus? Why do some of them end up in the ICU and die? And why some of them end up in the ICU and recover? And why some of them get infected but still remain asymptomatic? So that is the premise. And she touched a little bit upon how they are trying to collaborate, and also looking at children that develop some multi-inflammatory systemic features after SARS-CoV-2 infection. So there's a collaborative effort going on to try and resolve the unique variants versus the common variants, and to tease out this pathway, because there are so many other human genome initiatives that have started in 2020, right after this COVID-19 pandemic. So when you look at that, the amount of literature and information from last year alone on COVID-19 is absolutely phenomenal, right? It has just been exponential. But if you look at what are the variants that have been actually identified, what is the yield of testing hundreds and thousands of individuals, and also sequencing the SARS-CoV-2 genome along with the host genome, if you look at the sequencing yield or the sequencing output, in literature, it's really not a lot. That means there is a lot more that is hidden in the human genome that people have not interrogated. So this is just one example of that initiative. The other thing that I just to add on to what Erik was saying is I'm just going to cite one example in leukemias and lymphomas or hematological malignancies. One, there are two ways that the tumors start progressing when you look at like one class of structural variation. So either there will be a fusion protein, which is oncogenic in nature, which, one example is the BCR-ABL fusion, right? It's a fusion protein, oncogenic, and we know we have the wonder drug Gleevec for it. But there are some fusions that happen because of a translocation that bring in a proto-oncogene in the vicinity of a constitutively expressed gene. So one of those, for example, is the IGH translocations, which are very common. It's the immunoglobulin heavy gene, very well reported in all types of leukemias and lymphomas. And there are almost 40 translocation partners or more than that are reported. So it's very difficult for a laboratory that is working on hematological malignancies to try and tease out what is that translocation partner. Now, sequencing, just because of the polymorphism and the cluster and the number of these immunoglobulin genes, cannot pick up any of these translocations or any of these SVs. So this is where optical genome mapping, just because of the long molecules and because of the labels over there and the accuracy to be able to show those translocation partners. And all of this now is being published by multiple people, both in US and Europe and rest of the world. And this kind of like showcases the utility of optical genome mapping in certain classes and certain unique applications. Absolutely.
So Alka, we're over halfway. You're very passionate on this topic. This is AGBT. Have you said what you want to say? Erik said the movie has yet to play out. How do you see the movie?
I completely agree with Erik. And one of the reasons is, as I mentioned, karyotyping and has been in use since when? Like 1970s. The reason why it's been there, right? And then FISH evolved and standards and guidelines came in. Remember, there is no FDA approved or cleared for karyotyping or FISH. I mean, those assays are all LDTs that have been used since 1980s and 90s, right? For the most part, there are a few FISH ones for some applications, but the majority of them. And then came microarrays in 2010 that were adopted by medical associations such as American College of Medical Genetics, American Academy of Pediatrics, American Academy of Neurology. And those set the pace for what should geneticists, pediatricians, neurologists, pathologists, what should they do when they are faced in a situation when a patient shows up with specific clinical indications, and what testing should they follow for patient management and care. So this is what I have learned all along, and this is what I have been part of. And I've seen the evolution of, Sanger sequencing being used for single gene disorders or sequencing variants, and then moving on to next generation sequencing, again, around 2010 and so, and then exomes and now genomes. And every time a technology came in, it was like, well, we're going to take care of structural variations too, and these technologies may not exist or will be obliterated. But guess what? Cytogeneticists have evolved into cytogenomicists, which is what we call as a next generation cytogenomic revolution. However, we realize that all of these emerging technologies that are becoming the standard of care, if we look at even prenatal diagnostics, right? So the International Society of Ultrasound and Obstetrics and Gynecology or American College of Obstetrics and Gynecology, all of them have recommendations and guidelines for confirmatory diagnostic testing. And all of them cater to the detection of structural variations. So now if we look from 1970 to 2021, where we are, what is this era and what are we going to see now? We are going to see all of those hidden parts of the genome. We are going to be able to identify in terms of discovery and diagnosis. And I also believe that this is just the beginning of how structural variations and, optical genome mapping can be used as a tool for patient care and management. That is what we are here to do, right? For genetic testing, for betterment in terms of diagnosis, so that physicians, pathologists, oncologists can better manage their patients. So this is the reason why I've joined Bionano Genomics.
So Alka, you mentioned that, you joined the company to kind of push them along for certain things. Do you want to talk about that, like what you'd like to see happen in the next year or two, and also what you're hearing from labs like you used to work in and even around the world?
Absolutely. So as I mentioned, right, this optical genome mapping is a very unique technology, right? That it fills the void in so many places. Just one technology that can even enter the prenatal clinical application space to postnatal, to leukemias, lymphomas, and solid tumors. If you just look at, as I was mentioning earlier, the continuum of care, it just is an ideal technology. But there's more work to be done, absolutely, because every lab that wants a platform wants it to be cost effective, wants it to be ease of adoption, wants to be able to do the analysis and interpretation in a manner that that is understandable, right? A lot of the genome or genomics or information that comes out, a lot of algorithms need to put into place before you can make sense of all of that information. What we are trying to do here is to, provide these variants in a manner that the labs can understand it, relate to it, and can immediately understand the clinical interpretation or application of it. Now, in terms of what we are trying to do here, what I would like to see in the next, in this year and the next year is exactly what Erik was alluding to. We are going to be promoting and doing a number of clinical studies with a number of our partners and early adopters of our technology so that they can develop all of these applications. And now we will be directly working with them so that we hear their voice. What do they want in their lab so that we can help them do their job better? And no matter which way we look at it, do we need to develop more sample type applications? Do we need to, how much more throughput do we need to increase? Do we need to reduce the turnaround time? What would be the most cost effective way that they get all of this high resolution genomic information and it is affordable for them? And along with that, allow them to make sense of all of this data that they have never seen before. But they can look at, relate to, and say, I get it. I know what the structural variation means. I know I can see this translocation. I don't need 20 years of experience to sit on a microscope to look at a metaphase and say, I don't know what this is, and I've not been trained on it. But to look at optical genome mapping data on the screen, and it just tells you exactly what's going on in the genome, is something that, I think that is how we'll be able to help all of our customers. So this is what I would like to see this year. There's multiple applications by multiple laboratories, and just to, move forward in that direction.
Well, it's really fun to share your passion for this, and I look forward to hearing more about that, seeing that in the future. Erik, Alka, this has been a real pleasure. Thank you.
Thank you very much.
Thank you, Daryl.
And thanks to the folks at AGBT and LabRoots for making this a successful presentation. Thank you, everyone.
Thank you.